Hello everyone and welcome back to another edition of Bio 205. The next two chapters we'll be discussing the immune system. So to begin, what is immunity? Immunity is simply the ability to thwart off disease and protect against disease-causing environmental agents. There are two categories of our immune system. One is the innate or nonspecific immunity, which includes general defenses that are always present and can be used against pathogens regardless of type. Examples include barriers such as the skin or a response such as fever. The second type is the adaptive or specific immunity, and this includes defenses that are fine-tuned to a particular pathogen and can be retained for future encounters. Examples of this include specific cells, such as T cells that can be adapted to target a particular microbe. Our body's immune system can be divided into three different lines of defenses. The first line of defenses is to prevent pathogens from invading. The second line are those general responses after a successful invasion by a pathogen. And the third line of defense is that specific response after a successful invasion. Sorry for the typo, but for chapter 17, we'll be discussing the first line defenses and the second line defenses. And for the following chapter, we'll be discussing the third line of defense. So all of these components of these lines of defenses operate in concert with one another rather than independently. So let us begin our discussion with the innate defenses. So again, these are non-specific, so they can act against many different types of pathogens. They have no what microbiologists refer to as memory, so therefore they aren't finely tuned for an individual pathogen, but rather they're generalistic. And many of these defenses are found in both vertebrate and invertebrate animals, so they are ancient in terms of evolution. Okay, so let's discuss the first line defenses. The first line defenses all pertain to the skin or mucous membranes, and they include physical factors, chemical factors, and your normal microbiota. Let us look at the skin and mucous membranes more in detail. We have lacrimal apparatus, and these are areas for draining tears. We have the ciliary escalator in the throat. We have earwax. We have urine flow. We have vaginal secretions. And we have vomiting and defecation. All of these can play a role in helping to prevent disease. The skin and mucous membranes are composed of many layers of tightly packed cells. Skin is both dry and salty. Mucous membranes secrete mucus that pathogens must penetrate. Lacrimal glands in the upper eye socket produce tears and pass them to canals in the throat. Tiny hairs called cilia that line the esophagus keep mucus moving into the stomach, not the lungs. So that wraps up the discussion of physical factors in the first line of defense. Now let's discuss the chemical factors. To the right, you'll see a zoomed in image of the skin, which includes sebaceous gland and sebum. Sebaceous glands are very important in the first line of defense, in that they produce an oily and acidic substance called sebum. As we've learned in the past, acidity is one way to control microbial growth. And the oily film also helps in that it creates a layer of protection on the skin. Another very important chemical factor is lysozyme. Lysozyme can break down the cell walls of gram-positive bacteria. It's present in perspiration, saliva, urine, and vaginal secretions. Next is our gastric juice. And the gastric juice contains hydrochloric acid, which again makes an acidic environment, which helps to control microbial growth. Also, vaginal secretions, earwax, and urine are all mildly acidic. We don't generally think of our normal microbiota as being part of ourselves. However, it is extremely important in the first line of defenses. It works through many mechanisms. First, it competitively excludes potential pathogens. Next, they produce compounds that can inhibit the growth of other microbes. And finally, they promote the development of a healthy immune system. Here are two examples showing the importance of the normal microbiota. The first is taking broad-spectrum antibiotics, leading to inhibition or killing of vaginal bacteria. This can cause the overgrowth of the fungus Candida albicans, and this will lead to a yeast infection. The second example is taking another broad-spectrum antibiotic, such as clindamycin, which can lead to the inhibition or killing of your normal microbia in your GI tract. And this can cause a risk factor to a C. diff infection and pseudomembranous colitis or C. diff colitis. Therefore, doctors also typically recommend taking probiotics with an antibiotic in order to help restore your normal microbiota during the course of taking antibiotics. So that concludes our discussion of the first line of defenses, which includes physical and chemical barriers. Next, let's discuss the second line of defenses. The second line defenses are, again, general defenses, which become active after a pathogen has bypassed the first line of defense. They include formed elements in the blood, inflammation and fever, and a variety of antimicrobials. Formed elements include different types of white blood cells, also known as leukocytes. All leukocytes are derived from pluripotent stem cells. Recall that all of the cells in the human body share the same genetic information. Rather, it's how they use that genetic information that differentiates the cells into their different forms, their transcription and translation. Pluripotent stem cells can lead to the formation of myeloid stem cells or lymphoid stem cells. There's a lot to see in this picture, but what I want you to focus in on are the mast cells, eosinophils, basophils, and neutrophils of the myeloid stem cells. And from the lymphoid stem cells, I want you to focus on the monocytes, T cells, B cells, and natural killer cells. We'll get into a discussion later about these formed elements, but first I would like to share with you this article that reports the second patient that has been quote-unquote cured from the HIV virus that causes AIDS. Essentially, HIV attacks one of your white blood cells, and what these scientists did is they did a bone marrow transplant, which is where those pluripotent stem cells that white blood cells are derived from, and replaced them with a patient who did not have HIV. And through the use of using that in a combination of retroviral drugs, they have found a complete elimination or no evidence of the HIV present in the patient. Okay, so let's continue our discussion of the formed elements in the blood. All of these formed elements have different tasks, but remember that they all work in concert with one another. First are the neutrophils. The neutrophils are cells that can leave the blood, enter tissues, and destroy foreign microbes by engulfing them. Next are the basophils. The basophils release histamine, and histamine triggers inflammation and allergic response. Inflammation allows increased blood flow to an area and increases the temperature of an area, which helps fight off an infection. Next, eosinophils destroy parasites by attaching to their surface and releasing peroxides. Remember that free oxygen radicals are very harmful to cells. Monocytes can leave the blood, enter tissues, and become what we call macrophages, which can engulf foreign cells. Dendritic cells resemble nerve cells in that they have long extensions. They can engulf foreign cells and begin to initiate adaptive immune responses. Natural killer cells target abnormal or infected host cells and release perforin, killing them. T cells and B cells play a crucial role in adaptive immune responses, which we'll discuss in the next chapter. The dendritic cells, the natural killer cells, and T cells and B cells are all called lymphocytes. They are called lymphocytes simply because they are white blood cells that exist in our lymph system. Now let's discuss the process of phagocytosis, or engulfing of cells. Phagocytosis is the process of cell engulfing and destroying an invading microbe is called phagocytosis. The GIF to the right shows an animation of a neutrophil destroying a bacterium in the blood. So the bacterium is the diplococci there, and you can see it works its way, and there it goes. Wonderful. They use chemotaxy. So attraction of phagocytes to foreign microbes is done through chemotaxy. Next, there's adherence of the phagocyte to the microbe. And then the next step is ingestion through the pseudopod extension, which you can see in the little GIF there to the right. They have a special vessel called a phagosome. And so this intracellular organelle engulfs the microbe and then digests it. So the phagosome fuses with lysosome containing digestive enzymes, which destroy the microbe. Now let's discuss the lymphatic system. So in general, we're all familiar with the cardiovascular system. The lymphatic system is a similar system in the body, but it circulates lymph fluid through special vessels, tissues, and the red bone marrow where lymphatic cells are formed. Similar to the cardiovascular circulatory system, there are tiny lymphatic capillaries that merge into larger lymphatic vessels that feed into different lymph nodes throughout the body. Different properties that you should be aware of when it comes to the lymphatic system are that it has a unidirectional flow, it's linked to the circulatory system, it moves lymphocytes around the body, and the B cells and T cells are activated in the lymph nodes. And blood plasma can become lymph fluid and vice versa. Let's now move the discussion of second-line defenses to inflammation and fever. Inflammation is a local response to an injurious agent associated with pain, redness, immobility, swelling, and heat. Its purpose is to destroy, remove, or confine the injurious agent and repair damage. There are several steps to this. First is vasodilation. Dilation of the blood vessels is responsible for much of the redness and swelling and immobility and heat to an area. It leads to the migration and action of phagocytes. As the blood vessels swell, they become more porous, allowing phagocytes to migrate to these sites. Repair begins during inflammation and is completed after causative agents have been removed. Fever is a systemic response to an injurious agent associated with abnormally high body temperature, which facilitates second-line defenses and repair. Fever speeds up reactions, helping repair happen quicker. Other second-line defenses tend to be more effective at higher body temperatures. Moving right along, let's now discuss antimicrobials. To begin with, let's discuss complement system proteins. There are over 30 proteins produced in the liver and circulate through the blood. They enhance, or in other words, complement the action of white blood cells. Next are the interferons. These are proteins that are produced by cells that have been infected by viruses, and they stimulate phagocytosis in order to try to put an end to the viral cycle. Next are iron-binding proteins. So these transport and store iron in order to limit or prevent the ability of foreign microbes from accessing it. Iron is one of those limiting nutrients in the body. And next are antimicrobial peptides. Peptides are short chains of amino acids, and these antimicrobial peptides inhibit cell wall synthesis, they lyse membranes, and do other antimicrobial tasks. Many of these antimicrobial peptides are triggered by an infection. Antimicrobial peptides, also known as AMPs, are produced and used by different tissues and by our normal microbiota and differ in their spectrum and action. The usual mode of action is to disrupt the membrane. They, again, since they're part of the second line of defense, they're nonspecific, and many of them can target bacteria, fungi, and viruses. Next are pathogen recognitions, and these are nonspecific. They're called PAMPs, pathogen-associated molecular patterns. So these are very general components to microbes that are often foreign invaders. Monocytes, macrophages, dendritic cells recognize general molecular structures that are essential to survival and pathogens. Take a moment to try and think of things that might be unique to pathogens on the outer part of their cell walls that our cells do not have. You may have thought of such examples as peptidoglycan, flagellin, the LPS layer, and then other components such as viral DNA or RNA, which is unique. So phagocytes use pattern recognition receptors on their cell walls in order to recognize the PAMPs. PAMPs are always essential to pathogen structure and function, so they are not easily mutated in order to evade our PRR, or pattern recognition receptors. In the next slide, we'll show some examples of PAMPs, such as lipopeptides, flagellin proteins, and peptidoglycan. The PRRs are the green forks. So, in this image, you can see a phagocyte, and the green forks are the receptors. And we can see that we have receptors for lipopeptides, flagellin, and peptidoglycan. And when these receptors bind to those molecules, they send information to the nucleus, or the DNA, which allows for the transcription of certain genes that will then lead to translation of proteins that are used in order to attack those foreign invaders. On the inside, you'll see that at the phagosome, there might be a nucleic acid receptor that recognizes a specific nucleic acid, DNA or RNA, of a foreign invader. And all of this can lead to the production and secretion of antiviral interferons and other cytokines. Cytokines are secreted proteins that act as signal molecules to communicate information between cells. Some cytokines actually self-stimulate, so the cells that secrete them also are stimulated by that information. Other cytokines can travel short distances to other cells, and some travel across the body through the blood. So here are those three different types of cytokines. The autocrine cytokines stimulate the same cell that secretes them. The paracrines send signals to nearby cells, and the endocrines send messages that travel greater distances throughout the circulatory system. There are many different types of cytokines, but we'll concern ourselves with three broad categories. The interleukins are secreted by many types of cells, not just white blood cells, and they regulate many different functions, not just immune system functions. They, in regards to the immune system, can promote the development and differentiation of T and B lymphocytes and hematopoietic cells for blood cellular components. Chemokines recruit white blood cell leukocytes to a site of infection. Interferons are a nonspecific defense against viruses. They tell neighboring cells to stop making mRNA, destroy RNA, and slow or stop the process of protein synthesis. You can see how this could be effective in defending against viruses, because viruses must take advantage of cellular machinery to produce viral mRNA, RNA, and protein synthesis. So in this image, you can see a viral infected cell releases those cytokines, and it tells the neighboring cells to stop synthesizing mRNA, RNA, protein synthesis, or even tell them to undergo apoptosis, or cell death. So therefore, when that infected cell tries to spread itself, it cannot spread itself efficiently.
Okay, now it's time for checkpoint questions. Checkpoint 1. Name one way in which normal microbiota keep transient pathogens at bay. Checkpoint 2. An increase in which formed element in the blood would be associated with an asthma attack or anaphylaxis. Checkpoint 3. Fill in the blanks. Blank cells and blank cells are activated in the lymph blank. Checkpoint 4. What were the three major types of cytokines discussed in this lecture? And checkpoint 5. Fill in the blanks. Blank released by infected cells cause neighboring cells to undergo blank. Okay, those are all of the checkpoint questions and the end of the lecture. We'll see you next time. Bye-bye.